So I'd like to start with a brief description of what early health technology assessment is and how it can help innovators in both the nanomedicine space and in other areas of life science. So I think you'll all be familiar with this general cycle of, or stages of clinical development of medical products. Most of you, I'm sure, are also familiar with so-called valleys of death in medical product development. And one that's less talked about is the second valley of death, which can happen even after you get regulatory approval for a product. If, for example, payers don't want to pay for it, so there's no reimbursement, or even if they do pay for it, there's just limited adoption. And that usually happens because the value proposition for providers, patients, and payers is not as strong as it could have been. And so what EHTA does is use methods from health economics, epidemiology, and related disciplines to help assess the potential value of a new medical product under development in the hopes of trying to reduce the probability that you encounter that second valley of death by helping innovators to focus in on the use cases and clinical applications that have the strongest value propositions for different stakeholders in the healthcare system. And so in this specific study, we were focused on assessing the potential value of a new PD-L1 companion diagnostic in the non-small cell lung cancer context. And so this work was done in collaboration with Michel Meunier and his lab at Polytechnique Montréal and their spin-off company, Vega Bioimaging. It was funded by Enman. And we are the early health technology assessment platform based here at UBC Pharmaceutical Sciences in Dr. Larry Lin's lab. And so just so we're on the same page, just give a brief description of the clinical setting. So atezolizumab is a PD-L1 inhibitor that's approved by the FDA for two non-small cell lung cancer indications conditional on the PD-L1 expression status of the patient. And so the way this works is the patient with non-small cell lung cancer has their tumor biopsied. That tissue specimen is assessed for PD-L1 expression using a variety of potentially different tests, and that's classified as either high expression, low expression, or negative expression. And the FDA label allows for its use as a first-line therapy for metastatic NSCLC in patients with high expression, and as an adjuvant therapy for patients with stage 2 to 3A NSCLC if they're in the low or high expression groups, but not in the negative expression group. And so one of the problems with the existing standard of care is the limited accuracy of these PD-L1 tests. So the figure on the left there is drawn from a study where the Nordic QC took cores from 4 different non-small cell lung tumors that they had assessed themselves and send them out to more than 200 different labs, asking them to profile them for PD-L1 expression using whatever methods they wanted. And there was some variation in the results that came back. And so what we were able to do is use this data to estimate the sensitivity and specificity of the real-world mix of diagnostic tests being used in this area. And what we found is that they're very much tuned towards specificity. There's very few false positives, but there are a non-trivial number of false negatives. So these are patients that do actually have PD-L1 overexpression and could benefit from atezolizumab but don't receive the therapy. So there does seem to be room for improvement in the diagnostic, in a PD-L1 diagnostic by increasing the sensitivity. The question is, how much room is there and what's the value of that room? And that's why we did the EHTA study. So for this study, we built an early cost-effectiveness model. And I'm not going to go into the details of what that involves, but the basic logic of cost-effectiveness analysis involves modeling at least two different strategies. So one, the intervention. In this case, the intervention is the new and hypothetically more accurate companion diagnostic, and the comparator, which in this case is the existing mix of tests. And then you can, in both pathways, the test results inform the therapeutic strategy. And what we can do is then estimate the total healthcare costs in both strategies, as well as the total quality adjusted life years, or QALYs, experienced by these hypothetical cohorts under the different scenarios. And if you're not familiar with a QALY, it's a combined measure of length and quality of life that is used in many health systems and is a recommended outcome for economic evaluations of most healthcare interventions. And what we can do a lot of things with this. One thing we can do is calculate the incremental cost effectiveness ratio, or ICER. And that's it's a measure of value for money that is used by payers in many health systems, including the United States, to, as one input into their coverage and reimbursement decision making. In the United States, the general willingness to pay threshold, so the ICER value that will generally make payers think, oh, that's probably cost effective, maybe we should think about paying that, is somewhere between $100,000 and $150,000 per QALY gained. And so we built those two cost effectiveness models, and once we had those built, we could do a number of analyses. So first we did a headroom analysis, which basically assumed a hypothetical companion diagnostic with 100% accuracy and the same price as existing in vitro diagnostics. And the reason to do that is to look at what's the maximum potential value and health benefit that you could be adding with a new technology in that specific clinical niche. And what we found here is a result that the adjuvant indication has quite a bit more headroom than the metastatic indication. So having a more accurate diagnostic generated twice as much additional health, so 0.08 QALYs versus 0.04 QALYs per patient. And the ICER was around $49,000, which is far below the $100,000 per additional QALY threshold, whereas for the metastatic indication, it was close to $150,000, so close to people might, where people might decide that's just not cost effective. So a lot more potential value in that indication. So another thing we can do is then use the model to estimate a maximum value-based price, where you increase the price of the new diagnostic in the model until you hit that $100,000 ICER. What the price suggested by that analysis was $4,000 per patient. Now that's a lot higher than the pricing for existing companion diagnostics, and probably not obtainable, but it does suggest there's a lot of room to price a new diagnostic for that indication. So for example, if we took $500 per patient, that's at the upper end, but within the range of the prices for existing companion diagnostics on the market. Another analysis we can do is a threshold analysis to look at, well, how good does the test have to be cost effective at that price point? And so what we found is that if the test could achieve at least 93% specificity and sensitivity, it would be cost effective. Obviously, more accuracy is always going to make a stronger value proposition for payers and other stakeholders, but that doesn't need to be perfect, basically, to be attractive. And finally, we can use some of the inputs from the model to estimate the potential value to both the diagnostics manufacturer and the pharmaceutical manufacturer in this case. I won't go into details on, although I can tell you more in questions, but basically, yeah, based on general estimate, like we can say that for this indication, the potential size of the market opportunity for a diagnostics manufacturer would be in the single millions in the United States per year, and in the low tens of millions probably for the pharmaceutical manufacturer. And that happens because there will be new patients that are currently being classed as false negatives who would be identified and would be receiving therapy with a PD-1 or PD-L1 inhibitor in this situation. So conclusions, adjuvant indication has a lot more value headroom than the metastatic indication. There's substantial pricing room, and the test does not need to be perfect to be cost effective for that indication. And so that might point to a product profile that they might want to aspire to. So happy to answer any questions, and yeah, if you're interested in EHTA more broadly, please do reach out to us. We'd be always happy to talk. Thank you.
That was great, Nick. I'm wondering why more people don't do this before they start a project. So how is this work guiding the PI that you're working with?
Well, that I think probably we should leave that to, to him to
Okay.
answer. But we are planning on following up as part of the overall platform with some interviews later on as part of an evaluation to try to get a sense of that and publish those results eventually in a non-identifying way. But yeah.
Yeah, I was just curious if he's taking these numbers in the design of his assay that he wants to develop for the PD-L1.
I can't speak to
Okay.
that, but yeah.
Yep. Other questions? Yep.
What are the, okay, you state now pricing of like $500. What are the competitive products? Like what is the pricing? Just as a orientation.
Yeah, well, the existing IVDs are below 100. So in this the PD-L1 assays, like per patient. So this would be actually like more than 5x price, 5 times more than the existing test, and still looking pretty good from a cost effectiveness perspective. I'm not saying they could achieve that price necessarily, but there's room. Is the suggestion, but.